So in this video, we're going to talk about the bacterial plasma membrane, some things that make it a little bit different than eukaryotic plasma membrane. And we're also going to talk about different types of transport, including one type of transport across the membrane in particular called group translocation that bacteria use to transport sugars. Right? And so the plasma membrane of bacterial cells functions the same as it does in eukaryotic cells. The job of a bacteria's plasma membrane is mostly to separate the inside of the cell from the outside environment. You want to keep the cytoplasm or the inside separate from the world. However, that being said, the plasma membrane does have to allow some things to both come into the cell because bacteria needs nutrients that it gets from the external environment, and the plasma membrane also needs to allow some stuff out of the cell because bacteria make waste products just like you or I, and they need to pee or poop those waste products out. And so the plasma membrane of bacterial cells, while creating a barrier between the inside and the outside world, also needs to be selectively permeable, letting some things in and out and preventing others from moving in or out. The plasma membrane also has to function as a way for the inside of the bacterial cell to interact with the outside world, right? So as I already mentioned, the plasma membrane has to have a way to allow nutrients to come in, as well as to eliminate waste products. And the bacteria plasma membrane also has to have a way to receive signals from the outside world and then react to them on the inside of the bacterial cell, right? So it can sense what's going on in the world around it and adequately respond. Importantly, the bacterial plasma membrane has additional functions as compared to our plasma membrane in our eukaryotic human cells. Because bacteria don't have membrane-bound organelles like the mitochondria or chloroplasts or the endoplasmic reticulum, all of the processes that need a membrane in order to happen, such as cellular respiration and the Krebs cycle, photosynthesis, as well as synthesis of other cellular components like lipids and the cell wall, also all have to happen in the plasma membrane. And so in a bacterial cell, the plasma membrane is actually performing even more functions than it does in our cells, because that's really the only membrane available to bacteria, because they don't have any membrane-bound organelles. So just like our plasma membrane, the bacteria plasma membrane is a phospholipid bilayer, which means it's 2 layers of phospholipids. On the outside, it has a layer of hydrophilic heads. And then here, another hydrophilic heads with hydrophobic tails pointing inward towards each other. So you've got a phospholipid bilayer. Those phospholipids have those heads pointing outward and tails pointing inward towards the center of the membrane. And that allows the outside of the membrane to interact nicely with water on the outside in the environment, as well as the cytoplasm on the inside. And then inside of the plasma membrane, or that lipid bilayer, we generally see a series of embedded proteins. Some of those proteins might be involved in cell recognition. Some of them might be involved in being pathogenic. Some of them might be involved in transporting things across the membrane. But the lipids create the structure. The proteins will do the work that the plasma membrane requires. Respiration, transport, acquiring nutrients, eliminating wastes, all of that stuff is down to the embedded proteins. Right? And so membrane lipids, as I mentioned, or phospholipids, are what we call amphipathic because they have 2 different chemical properties on either side of the same molecule. So here's a picture of your phospholipid. This part here is called the head. It is polar and hydrophilic, which means it loves water. And then these 2 tails coming down here are nonpolar and hydrophobic, which means that they fear or do not like water. And so because of this amphipathic structure, the most natural, spontaneous thing for a bunch of phospholipids to do is form a phospholipid bilayer, just like the plasma membrane. And so in water, which is pretty much what all of the environment as well as the inside of our cells is composed of, phospholipids are going to self-assemble into that phospholipid bilayer structure, where the heads are pointing outward towards the environment and inward towards the cytoplasm, and then all the tails are pointing towards each other. So in a plasma membrane, the lipids on either side of the two leaflets or the two sides of the bilayer are different from each other. And so on the outside, you see a lot of here red and blue lipids, whereas on the inside, the part closer to the cytoplasm, there's a lot of green and lime green lipids. And that's just down to different functions of either side of the bilayer. Structure determines function. And so depending on what's going on in the structure of the plasma membrane, it might have different lipids on the outside layer versus the inside layer. And the lipids in a bilayer can actually move around. They're extremely dynamic. And so the phospholipids on this layer here can move front to back. They can move side to side. They can actually spin in a circle or have rotational motion. And they can also flip from one side of the bilayer, say the side facing the environment, to the side facing the cytoplasm. Lipid composition, depending on what type of lipids you find in a membrane, can affect how dynamic or how fluid a membrane is. And so on the left, you can see an example of a fluid membrane. And on the right, what would be considered a more rigid or stationary membrane. And so what you'll notice in the fluid membrane is that there's a lot more of these unsaturated fatty acids or these kinked chains in this membrane, and that increases its dynamicness or how fluid or how easily it changes. Whereas in this one on the right, there's a lot more of the saturated fatty acids, which makes a more stable membrane. And it's good for membranes to have both fluidity and stable areas because membranes need to do a lot of different stuff. And so some of the areas that have more stability are called microdomains. And you can see them here in green in this picture on the bottom. And in the microdomains, there's very little movement of the lipids. There's not a lot of rotating or flipping back and forth. Those lipids are stable because those areas, microdomains, are holding important proteins together. In this case, proteins that allow stuff out of the cell. In this case, maybe proteins that are involved in a signaling pathway. And bacterial plasma membranes contain some different lipids than eukaryotic cell membranes do. Eukaryotic cell membranes contain a lot of the molecule called cholesterol, which you can see here on the top. Cholesterol is a pretty big and bulky molecule, and so it tends to increase the fluidity of membranes in our cells as humans. And bacteria don't have any cholesterols in their plasma membrane, but what they do have is something that looks a lot like it. And so if you look down here at this molecule, to the untrained chemistry eye like mine, these two molecules look very similar. And these molecules that bacteria have that are kind of big and bulky are called hapenoids, and they help to increase the fluidity or the dynamicness of the membranes in bacteria. So you will never find a hapenoid in a eukaryotic cell membrane, and you will never find a cholesterol molecule in a bacterial cell membrane. They are exclusive to their different types of cells, but they serve a similar function in increasing the fluidity of the membranes. And so bacteria need to take things in from the environment as well as eliminate waste products, right? They need to take in certain elements like carbon and hydrogen and phosphorus, as well as small trace elements, some of the metals that they use to power their enzymes, and growth factors, stuff that can't be made by the bacteria, they need to get from the environment. And that means that there are mechanisms in the plasma membrane in place for them to transport nutrients in and waste products out. And so you have likely discussed some of these before in intro bio, different types of diffusion, both passive that doesn't require energy and facilitated, which does, as well as active transport, which is transport that usually is powered by energy like ATP. We are not going to discuss those today because you should have had a pretty nice background of them from intro bio. But I do have another video that you can check out that has descriptions of all of those if you need a refresher. What I am going to cover is group translocation, which is a bacteria-specific type of transport that they use to transport sugars from the outside environment into the cell so that they can use it for energy. And so group translocation is movement of molecules from an area where their concentration is low to an area where their concentration is high. So this is a low to high concentration movement. And because it's low to high, it's technically movement of molecules against their natural concentration gradient. So this is going to require some energy input. Anytime you want to move a molecule from an area where there's a little bit of it to an area where there's already a lot, it's going to require energy input. And so the thing that's special about group translocation is that as the molecules are coming into the cell, they get chemically modified or changed. And as I mentioned, this is going to require some input of energy. And the energy input here, rather than ATP, which is an energy currency we're familiar with, is PEP, or phosphoenol pyruvate. And so the PTS system is a sugar transfer system that uses PEP for energy and modifies sugars as they come into the cell. And I'm going to explain how that works on the next slide. Right? And so you hear PTS transport is moving different sugars into the cell, and the chemical modification that happens to them is phosphorylation, or adding a phosphate group. And so the first thing that happens is that phosphate group has to move from PEP, which has a phosphate on it, to a carrier enzyme called enzyme 1. And so here you can see PEP donating its phosphate to enzyme 1, and here enzyme 1 now has this P or phosphate group on it. And the chain of passing the phosphate continues. Enzyme 1 is able to pass its phosphate to heat protein, or HPR. And then heat protein transfers the same phosphate to enzyme 2, which you can see here. Enzyme 2 is a complex of proteins that's inside of the plasma membrane. You can see enzyme 2 for mannitol, which is one sugar, up on top. This is enzyme 2 for glucose on the bottom. And so HPR will transfer a phosphate group to those enzyme 2s. And then enzyme 2 will take the sugar and transport it across the membrane and phosphorylate it in the process. So mannitol is coming in. As it does, this phosphate group is passed to mannitol, and it now becomes mannitol phosphate. Glucose is coming in. This phosphate group is passed to it, and it becomes glucose 6-phosphate, right? And so this whole transport system relies on the transfer of phosphate groups from PEP to enzyme 1, to heat protein, and then to enzyme 2, and finally to whatever sugar is being moved into the cell. And enzyme 1 is shared for every single type of sugar that's moving into the cell via this mechanism, but there's a different enzyme 2 for every single sugar. Mannitol has an enzyme 2, glucose has an enzyme 2, fructose, sucrose, dextrose. Every single sugar will have its own enzyme 2, and that is why down here it says enzyme 2 is what gives this transport its specificity, because there's a specific enzyme 2 to transport each individual sugar.